embryonic stem cells are taken from a pre-implantation stage. These embryos are then placed in a petri dish containing a nutrient broth. Mouse embryonic stem cells are treated so they do not divide. These cells are used to allow the human embryonic cells to stick to the bottom of the dish. The mouse cells are used as feeder cells in order to provide nutrients to the broth.
There are a couple of pros to growing stem cells from human embryos. One of these pros is that embryonic stem cells can be grown into virtually any type of cell. Embryonic stem cells are also only discarded embryos and are never derived from eggs that are fertilized within a human. These embryos do not have the capability to feel pain or emotion, so there is no trauma to the embryo.
In the current realm of biotechnology, scientists are on the verge of refining the ability to grow embryonic stem cells. This emerging technology should not be embraced by society because there are more cons to this process than pros.
There are many cons to this process. The first con is that an embryo meets the technical definition of life. According to the Biology Online Dictionary, a distinctive characteristic of a living organism from dead organism or non-living thing, as specifically distinguished by the capacity to grow, metabolize, respond, adapt, and reproduce. An embryo meets all of these qualifications. This process is also inefficient and time-consuming, with a minor amount of success. There is no guarantee that rejection of the cells will not occur. Human embryonic stem cells could also potentially transform into something more serious, such as cancer.
Some people think this is a good option for repairing injuries or possibly saving lives. However, there's a whole realm of stem cell options besides embryos that can do the same things. There's more research done on other types of stem cells, and there are more cons than pros with using embryonic stem cells. There are other types of stem cells, such as iPS, that do not cause a rejection within the body, but embryonic stem cells could, which means other types of cells would be safer to use.
In conclusion, we do not think that embryonic stem cells should be used in research. They are a safer alternative to these stem cells, and we think that although scientists can learn a lot from researching these embryonic stem cells, there are too many risks involved with growing them for human use at this time. We do not think that this emerging technology should be embraced by society.